Hello everyone, my name is Ryan Davis and I'm a senior here at Marsville University and I have a major in zoology, a minor in chemistry. And in lieu of me doing a video, here's a picture of my face so you can still see who I am. I conducted my research on the protective properties of plant extracts on Saccharomyces cerevisiae under stress. And I want to give a shout out to Dr. Griffin for being my mentor on this project. He was super helpful, couldn't have done it without him, so thank you. So what causes cellular stress? There are three main things. There's reactive oxygen species, also known as ROS, high temperatures, and toxins. ROS is a natural byproduct of cellular respiration, which occurs in the mitochondria. And antioxidants are within the cells and they suppress the effects of ROS. However, when ROS is in abundance, the cells are overwhelmed and the antioxidants can't combat them, leading to cellular damage and then ultimately cell death. High temperatures move cells outside of their optimum range, denaturing proteins, causing them to lose their function, and then leading to cell death. Toxins, they inhibit enzymes within the cells, undermining a lot of the cell's processes, causing the cells not to function properly, leading to cell death. For my research, I focus mainly on ROS, particularly because it is prevalent in the onset of many diseases like Alzheimer's and cancer. My model organism for this research is Saccharomyces cerevisiae, also known as yeast. The reason why I chose this is because you can control whether or not they use their mitochondria based on the food source that you give them. With glucose, they don't have to use the mitochondria. With acetate, they do. And like all the other organisms, they have antioxidants to combat ROS. But if the ROS is in abundance, they can't use that. So where are they going to get the antioxidants from? Well, they're going to get them from these plants. So I have the Rhododendron maximum on the left, which is known as the great laurel, and the Kalmia latifolia on the bottom right, which is the mountain laurel. And both of these plants have antioxidants, which plants produce as a secondary compound to combat herbivory. And the main two antioxidants present in both of these plants are phenols and flavonoids. So here's my thesis statement. Rhododendron maximum and Kalmia latifolia extracts will protect Saccharomyces cerevisiae from heat damage and oxidative damage. So here are my methods. Rhododendron maximum and Kalmia latifolia extracts were made from the leaves collected from both of these plants on Buffalo Mountain in Johnson City, Tennessee. These leaves were ground in a mortar and pestle, soaked in 50% ethanol to extract the organic compounds, then placed in a rotary evaporator overnight. Then the residue from the evaporator was placed into solution and then sterilized so we could use them for the yeast cells. And I ran three main tests: heat shock, oxidative stress with hydrogen peroxide and iron sulfate. In each test, there were three groups: the treatment group with cells in stress, cells with only stress, and cells with no stress. And each of these tests were ran in three replicates of threes for each experiment. And then, along with those three replicates, I repeated each of those with the cells grown in glucose and the cells grown in acetate for the mediums. To analyze the data that I gathered, for yeast cells, there were multiple assays that were ran. For the methylene blue viability assay, you would stain the yeast cells in methylene blue solution. And living cells could easily diffuse the dye and would appear clear, as you can see in the picture. But the dead cells would stain blue. So I would go through and count all the cells and then calculate the percent viability from knowing which ones are alive and which ones are dead. For the spotting viability assay, I would do a series of serial dilutions to measure the concentration of at which the yeast cells are still able to grow without being inhibited. For a statistic analysis, I use one-way ANOVAs followed by Fisher's post hoc test and a paired t-test. So before I did anything with my research, I had to ensure that the extract was not toxic to the cells. So as you can see in the graph, I was measuring the concentration of the yeast cells in solution when they were grown without extract and with extract. And there was no significant difference. And this is what I wanted to see, so I could continue on with my research. So the first test I did was heat shock with mountain laurel extract and solid YED. And YED is glucose. And then also did another test with heat shock with rhododendron extract and solid YED. So what I'm looking for in these graphs are for the heat and the extract group to look more like the no heat group. Because if it looks more like the no heat group, then that ensures that the plant extracts are protecting the cells. As you can see on the left of the graph, it shows the percent viability. It will show that it's been living longer and surviving. If the heat and extract looks more like the heat only group, then it shows that the extract is not effective and it can't protect it. So with the mountain laurel extract, there was no significant difference in between the percent viability with the groups, meaning that the extract, it did nothing for the cell's protection. On the other hand, on the right side, on the right graph, the rhododendron extract had a significant effect in protecting the cells from stress. So the rhododendron extract increased average percent viability. And with the previous test, I only had time constraints, so there was only so much I could do, so I had to choose between which extract I would use, and the rhododendron extract had significant effects, so I focused on that. And then I was growing those on solid cultures. And those solid cultures I have to refrigerate, but as I was refrigerating them, the cells were getting older and they were more susceptible to stress. So to prevent that, I started growing them in liquid cultures, so I have new cell growth every time. So I had to repeat the heat shock test with the rhododendron extract. So on the left graph, you can see that in liquid glucose, that the cells were protected from the stress with the extract. However, on the right side, when it was in liquid YEAC, which is liquid acetate, the extract cannot protect the cells, and there's no difference in between the groups. So rhododendron extract could only protect against heat damage in liquid YED cultures. The next test I ran was the hydrogen peroxide oxidative stress test. And on the left graph, when they were in the liquid YED, there was no significant difference. But when they were in the liquid acetate, there was very significant difference. And this shows that the extract was able to protect the cells from oxidative stress. And this also shows that the yeast cells are more susceptible to oxidative stress damage when they're using the mitochondria as compared to when they didn't have to use it in the glucose. So then these are the spotting assays. When looking at these assays, you're looking for the difference in between colonies and to ensure that the extract didn't inhibit growth and that there was no cytotoxic effects. And this one is in the YED plates with nothing else added. And then you can see there's no real difference in between the colony growth with or without extract. So that's what you want to see. There's nothing bad there. When grown in acetate plates, there's some slight differences between when each experiment when they were grown with extract and without, but not enough to really, focus on to really like nothing out of the ordinary to be clear. Next, I did the iron sulfate oxidative test with the spotting assay and YED. As you can see, there's no real difference again, so you can write that one off. And then this one is the iron oxidative stress test with acetate in the spotting assay. There's slight differences, but still within the range of ordinary, so you can't really say anything was going on there. So the important takeaways from this research that I've done is that the rhododendron extract was significant, had significant antioxidant effects in protecting the yeast cells from stress, and there were no observed cytotoxic effects in any of the experiments that I did. So the extract is a good reducer of the effects of stressors. And with more research, there is a great potential to have this be used as supplemental therapy for diseases involving oxidative stress that I mentioned earlier, like Alzheimer's and cancer. And this is a good alternative to those synthetic compounds synthesized in lab, since these are more natural biotic compounds. Here are my sources. Any questions? If you have any questions, you can email me. My email is in the first slide. It's ryandavis1@mhu.edu. So let me know. I would love to get back to you. And if not, thank you for watching, and I hope you have a wonderful day.